Welcome to Propellacure's continuing series of interviews with leading experts in the inflammatory bowel disease research field. My name is Angela Clark, and I'm the president of Propellacure. And we're really excited to have today our special guest, Dr. Vincent van Unen, who is an assistant professor of immunology at the Leiden University Medical Center in the Netherlands, where he leads the Vincent van Unen Research Group. Dr. van Unen is an expert in mucosal immunology with a focus on immune-mediated inflammatory disorders. His lab aims to dissect key immune mechanisms driving disease with the goal of improving diagnostic precision and developing more effective treatments for chronic intestinal conditions like IBD. Several years ago, Propellacure, in partnership with the nonprofit Cure for IBD, awarded our very first grant in support of a groundbreaking study that was recently published, which we will be discussing today. The research for this report was led by Dr. van Unen's research group at Leiden in collaboration with Stanford University's School of Medicine. It is titled Shared CD4+ T-cell receptor specificity groups in Crohn's disease and ulcerative colitis. So we're going to learn a lot today. Dr. van Unen, it's so nice to have you with us. Congratulations on the publication of this important study in JCI Insights.
Great. Thank you, Angela, for your kind introduction. And of course, I'm very grateful for the support of Propellacure to make this study possible. And of course, all the patients that were involved. And I'm really looking forward to discuss the content of this research with you in the coming hour.
I think everyone's going to be really interested to hear what you have to say. We're all excited about it. Before you go in and start your presentation, what questions were you hoping to answer when you first began this project?
The real motivation behind this study was the fact that we know that there were certain genetic risk factors involved in IBD, such as HLA that presents antigens, and we know that T-cell responses are involved in IBD. However, these genetic risk factors and these T-cell responses haven't really been studied together. So the real question I wanted to ask is how does these genetic risk factors within your HLA shape your T-cell patterns? And how similar or dissimilar is that between Crohn's disease and ulcerative colitis? So that was really the big question to ask, which of course would set the stage for trying to identify triggers that drive the disease.
And I know that's been a big question in the research community and a big question for patients. So I'm going to turn it over to you. I know you have some slides to show us, and we'll take a look at those and then take a listen.
All right, wonderful. I'll just go on to share my slides with you. All right, do you see my slides?
Yes.
All right, wonderful. Yeah, so as we mentioned, is that I'm very interested in understanding how genetic risk shapes your T-cell recognition in inflammatory bowel disease. And that was really the aim of this study. And here I outlined, why does this matter? Well, we know that, of course, Crohn's disease and ulcerative colitis, they differ clinically, but they do share certain features. For example, both diseases involve these dysregulated memory T-cell responses. In addition, both Crohn's disease and ulcerative colitis also share risk factors in the HLA molecules. Now, why is this interesting? HLA are the peptides that are presented by your antigen-presenting cells. So these HLA molecules present antigens to your T cells. And T cells, they express a T cell receptor, or TCR, which is a very unique feature of T cells because these are like a unique sensor that screens for antigens to assess whether this is potentially harmful for our body or not. So this interaction of antigen-presenting cells with T cells is very important in mounting an immune response. So since HLA shapes antigen presentation, we really wondered whether this mechanism would leave an imprint in your T cell receptor repertoire in the context of IBD. So to address this question, we linked the HLA genotypes of patients to their T cell receptor repertoire. So we analyzed over 3.1 million T cell receptors in both Crohn's disease and ulcerative colitis patients and compared them with healthy controls as well.
Dr. van Unen, what technology enabled you to compare those vast numbers of signatures?
Right, yes. So that's indeed a technological innovation that in Mark Davis' lab at Stanford University, they're really an expert on. So these are high-resolution sequencing approaches. So basically, from the memory T cells from a patient, we can, in a high-resolution sequencing, characterize for each T cell their T cell receptor. So this is based on next generation high-resolution sequencing. And in terms of the HLA molecules, this is also a high-resolution genotyping so that we ensure that both the T cell receptor and the HLA are very accurately measured in these patients. All right, so in total in this study, we had included 33 patients with IBD. 20 with ulcerative colitis and 13 with Crohn's disease, and we also included 14 healthy controls. Now you can imagine when you have 3.1 million T cell receptor sequences, it involves computational approaches to make sense of that, because we wanted to understand what are some unique patterns in the T cell receptor repertoire in patients versus healthy controls. So for that, we used an advanced bioinformatic tool, which is called the Glyph2 algorithm. And basically what this algorithm does is that it clusters the T cell receptors by screening their antigen binding motifs that are likely the motifs that recognize the antigen that are being presented in the HLA. So when we group these similar receptors together that may recognize the same antigens, now we only had 460,000 groups left, which is of course still a lot to go through. But there we had a very robust filtering procedure. We wanted to be ensured that these were statistically relevant. And most importantly, we were interested in the T cell receptors that belong to memory T cells, because it's the memory T cells, those are the ones in our body that have seen antigens before. So when we ensured that these T cell receptor groups that we found were enriched for the memory T cells and connected to their HLA genotypes of the patient, we ended up with 440 high confidence T cell receptor groups in this analysis. So then, of course, we were very curious to understand, is there a difference between patients and healthy controls for these 440 T cell receptor groups? And that is what we plotted here. Out of these 440 groups, 15 were significantly different between healthy controls and IBD. And here you see them plotted. So we found 5 T cell receptor patterns that were enriched in IBD patients, and we found 10 T cell receptors that were depleted in IBD patients. So this was a very exciting result for us, because this was giving us a clue that these 5 T cell receptor patterns that were associated with IBD are probably the ones that we should focus on, because these might be the T cells that are involved in a pathological immune response in IBD. So we wanted to understand this in more detail.
And this was totally new information. We had not seen this before in research, correct?
Right, because no one before has linked the T cell receptors to the antigen-presenting HLA molecules in IBD. So this was a complete novel finding, that in patients that have a genetic HLA risk factors, that we could find the T cell receptors linked with those HLA molecules was completely new. So this has not been investigated before. So of course we were very excited about this finding, and we wanted to, try to understand this in more detail. And I think this was one of the, very surprising finding for us, is that when we compared those 5 T cell receptor groups that were linked in IBD, first of all, we see indeed healthy controls do not have T cell receptors that belong to these groups. And we find them in both Crohn's disease and in ulcerative colitis, because here you see all the individual patients plotted. In many patients, we found these T cell receptors, and these T cell receptors did not discriminate between Crohn's disease and ulcerative colitis. So this suggested to us that despite that we see clinical heterogeneity among Crohn's disease and ulcerative colitis patients, in terms of the T cells involved, it may suggest that they have more similarities than we previously thought, and that they share an HLA-linked antigen recognition programs. And also, this was not one IBD T cell receptor clone that we found. We found that these T cell receptor specificity patterns were shared across multiple patients. Yes. All right, so then we took a further look into these T cell receptors, because as you know, we have also connected this to the HLA risk alleles. And here you can also see that those 5 T cell receptor groups that we found in the IBD patients were linked to risk alleles of patients. So for example, we found the DRB1-1101 HLA molecule, the DRB1-701 HLA molecule, the 1501, and the 1201. These HLA types are risk factors in IBD, and these were linked to the T cell receptors that we found enriched in the IBD patients. And if we then look into the healthy individuals, because there we also found 10 T cell receptor groups, you see that the majority of these are indeed also not connected to risk alleles. So this confirmed to us that there is a connection between the genetic susceptibility of IBD patients in their HLA molecules and the antigen recognition patterns, being the T cell receptors in memory T cells.
Now, you may have this information further along in your presentation, but is there any idea at this point what specific antigens the T cells may be reacting against or responding to?
Yeah, that is, of course, a big question, and I will touch upon this a bit later. But I can indeed already tell you that we have found antigen leads that were connected with these T cell receptor patterns, and they include 2 fungi, a Candida and the Saccharomyces species, and also an influenza A antigen from the M1 protein. So we have some antigen leads that we think could be the antigen that these T cell receptor recognize. And this is indeed exactly what we are following up on now, to see if this is really the case and whether this is the complete picture or just the tip of the iceberg. So I will discuss that in a bit further along in more detail. All right, so of course, having found T cell receptors in IBD patients that may be linked to an antigen responses, that may also have implications for, is IBD then considered an autoimmune disease, or is it an inflammatory reactive disease? And this is, I think, a big debate in the field that we still haven't really resolved. That more research is needed, but we wanted to also shed some light on this, because we have looked into more details at the phenotypes of those T cells where we found those specific T cell receptors. And what we noticed is that in a subset of these patients, so not all IBD patients, but in a subset of the patients that carry these HLA risk backgrounds, we found a very interesting subset of T cells. These are cytotoxic memory CD4 T cells. So here you see that these cells right here, they express granzymes, granzyme A, granzyme B, perforin. These are cytotoxic molecules. And when we looked at whether those T cells were clonally expanded so that they had seen antigens, we indeed see that it is right in that area where we find a lot of antigen experienced T cells. So this suggests to us that it's not only specific T cell receptors that we found, but also the T cells themselves carry a very pro-inflammatory cytotoxic phenotype that is considered, has also been considered more in autoimmune contexts. Then we looked in more detail at the T cell compartment, because there are many flavors of T cells. You have the CD4 T cells that we just discussed, but there are also CD8 T cells. And research from Mark Davis' lab at Stanford has previously shown that the presence of these regulatory KIR CD8 T cells are associated with multiple autoimmune diseases. And these are like, for example, multiple sclerosis or arthritis, these type of classical autoimmune diseases. These regulatory KIR CD8 T cells are elevated. And what we found is also that not for all IBD patients, but like for a subset of IBD patients, we found also quite an abundance of these regulatory KIR CD8 T cells. So this is another clue that suggests that, IBD is likely representing a spectrum where you have a more autoimmune part of the spectrum and a more chronic inflammatory perspective of the spectrum. And as a subset of patients may be more linked to the autoimmune side of the disease.
That's so interesting, because I know that whole autoimmune factor has been a subject of debate as well for a number of years now. So that it just seems very diverse, just patients representing all these different presentations. So quite interesting.
Yeah, absolutely. And, we already, of course, know from the clinical profile of IBD patients and their responses to treatment that it's really not a one-size-fits-all kind of disease. There is a lot of heterogeneity among, the symptoms and the treatment responses that patients experience. So what we see in terms of mechanistic basis of the disease is that this also seems to reflect that there is a heterogeneity. based on the data from this research, but also from other research groups, it suggests to me that it's not going to be one shared mechanism of disease among all patients, but it's likely going to be different types of mechanisms separating different subsets of patients. So I think this is also what this slide represents, that I think that this type of research that we've done here really support a path towards this antigen discovery, but also towards, a mechanism-guided stratification of IBD patients. This is like summarizing the main findings and approaches from our study. We had this big data from patients. We looked at all the similarities in their T cells. We had clever computational approaches to really focus on the most relevant patterns. And I think that this findings show that there are HLA-linked immune circuits, if you will, that could drive the disease. And this would open the door for more precise and indeed a need for more personalized patient care by linking your genetics, your HLA profiles. This could point for those patients that carry these risk factors, potential antigens that may be triggering the disease, and it would reveal which types of patients, which immune pathways are active. And I think when we start putting those puzzle pieces together, it could enable us to stratify patients, to group patients of IBD in more mechanism-based subsets. And that might allow for a more personalized and better treatment choices for patients. And in addition, I also think that how we traditionally look at IBD in terms of labels of ulcerative colitis and labels of Crohn's disease. And while for certain clinical features, these are, appropriate labels to carry, but when it comes towards, really the immune mechanisms that drive disease in these patients, there may be more similarities between UC and CD for subsets that carry, for example, these HLA risk alleles. So I think we should also really have an open mind in how we define the patient groups and whether we should move towards understanding more the mechanisms underlying the disease. And I believe that if we do that in the coming years, even in more details, that would allow for more precision care in IBD. So I think that is a bit the framework on how I view IBD from a mechanism point of view.
I think that's an encouraging thought because, taking a further research into Crohn's disease can yield insights into UC and vice versa, I think, in the long term. So what sort of testing do you think might arise from some of your findings in order to guide these precision type treatments?
Yes, so there are two active research projects that our lab is now pursuing based on this study. On the one hand, we really want to push the antigen discovery further. Based on their T cell receptor profiles, we now have some clues towards which T cell receptors we should focus on. And as I mentioned, there are some clues that maybe some fungi, such as Candida Saccharomyces, are involved. Maybe influenza A, like the flu, is involved. But we cannot really ascertain that now because we need, therefore, to really clone these T cell receptors in our lab, study it in the lab by feeding those antigens and studying whether indeed these T cells respond to that, and whether that is enough to drive pathology. So that is what we're now really looking in more detail for the antigen discovery. And as I mentioned, I think it's really only the tip of the iceberg. So we are now connecting our data with data from other research groups to have a much more in-depth understanding of antigens that may be driving those disease. And then indeed, secondly, our research is focused on better stratifying IBD by grouping patients more based on whether they share the same mechanisms. And for that, we are also now doing much larger patient cohorts to really try to understand, can we, by combining the genetics, the antigenic profiles, the T cell receptor profiles, immune pathways, when we look in this in more detail in larger patient groups, can we indeed propose more mechanism-based stratification of patients? So I think those are really like two ongoing research efforts in our lab, that this study that was kindly supported by Propel Cure and by the patients made possible, that I think could really push the field further. Then in terms of my presentation, I of course wanted to spend time to acknowledging the people involved. There's a lot of researchers involved in our immunology department at LUMC, our IBD specialists and other researchers at LUMC. We benefit also from computational tools that we developed. And this research was also made possible by Mark Davis at Stanford University. We really want to highlight Joshua Chan, who was the first author of this work, who was really driving this research, and all the funding that made this possible, especially, of course, Propel Cure, and all the patients that were involved in these studies. We are, of course, very thankful for.
Thank you. Well, this was has truly been a global effort with many people involved, different institutions, and we're just encouraged by what you're doing. Do you, taking this further, you alluded a little bit to what is next for your lab, some of the things that you're looking at. Can you expand on that further? And can we have any guesses as to what types of treatments might somehow address these issues someday?
Yes, certainly. Yeah. So again, I think it is really important that we start looking at IBD on a, per patient level, like in a personalized manner, also for obtaining mechanistic insight. And if indeed it turns out that patients that carry these HLA risk alleles that share also T cell receptor patterns, if we can substantiate this more in larger patient groups, and if this indeed we can demonstrate in the lab that this specific T cell receptor responses recognizing specific antigens in these patient subset, I think this would be very exciting because it gives us a way to better group patients and better understand mechanisms of the disease. I think it could also shed light in potentially what caused the disease in those patients. Was this indeed the initial trigger that resulted in the disease onset? So I think if our research points in this direction, but much more is needed to show that perhaps this is also related to what caused the disease in the first place. So then if that all were to, pan out, I think this would have indeed therapeutic implications. One, if we can pinpoint the bugs that were responsible for this, we could have preventative treatments of trying to prevent exposure to these bugs. You could have specific bacteriophage treatments that can very selectively eliminate certain bugs from your microbiota composition. Thirdly, if we know exactly which T cells with carrying these pathological T cell receptors were responsible for driving the disease, you can also think of ways of treatments that selectively eliminate those pathological T cells from your repertoire, while leaving the T cells intact that have nothing to do with the inflammation. And I think that would be really a game changer, because currently, we try our best to dampen the inflammation, and perhaps we dampen a bit, too much, and also the good cells, because many of the T cells in your body have been educated to, induce tolerance and to respect your body. And preferably, you want to leave those intact. And I think the current treatments, may not be adequate enough in discriminating the pathological T cells from the good T cells, if you will. So I think if we can really substantiate this and pinpointing these are the pathological T cells in the repertoire of patients that carry these HLA risk molecules, that I think it could pave the way forward towards new treatments that are much more precise, much more specific in only eliminating those T cells from your repertoire. And I think that's really, for me, like the dot on the horizon that we move towards.
That would be amazing. I know a lot of patients would agree. Now, I'm going to step back just a minute to ask you. You described influenza A as something you had possibly linked to all this activity, and a couple of classes of fungi. Can you tell us more about what you saw there?
Right. So basically, let me maybe shed some light initially how these bugs and these fungi and these viruses came on our radar. So once we knew the T cell receptors, those 5 T cell receptor patterns that we believe are, play a role in IBD, we tried to match those T cell receptor patterns to all publicly available annotated T cell receptors. So there are publicly known T cell receptors defined, including the antigen they recognize. And then we found that those IBD T cell receptors were bearing similarities to T cell receptors that have previously been demonstrated to respond to influenza A or to Candida, especially Candida albicans, or the Saccharomyces cerevisiae. So it's a difficult name for me to pronounce, but it's another fungi. So basically, at this point, we have only observed the T cell receptor. A very similar one has previously been described. So I would emphasize that this is therefore still quite, unsubstantiated result. I can't really at this point say, then therefore these are the ones responsible, but it gives us some clues. Now, the reason why I am still a little bit reserved in saying that this is now the ones to focus on, if you look at these public annotated T cell receptor databases, they are heavily skewed towards viruses and fungi. And there is currently a lack of annotated T cell receptors towards intestinal microbiota dominant species. So therefore, I think this is the tip of the iceberg, and I don't believe this is the whole picture. And so we definitely are currently taking these influenza, Candida, Saccharomyces along in functional testing and showing in the lab, do these T cells from IBD patients really respond to these antigens? And is this enough to drive an inflammatory response that leads to tissue damage? This is still to be determined, that we are actively pursuing. And at the same time, we are trying to bridge that gap of a lack of T cell receptor annotated antigens from intestinal microbes. So we are currently expanding our knowledge on T cell receptors that are linked to intestinal microbiota species. And I think once we have that important puzzle piece available to us, I think we will be in a much better position to really narrow down these are likely the antigens that play a role in IBD patients that carry HLA risk induce. So that's a bit where we are right now. Unfortunately, it's still early research. I cannot make any, strong statements that, we're there. But I think this study was such an important stepping stone for us to really start addressing this mystery of what drives IBD.
So like you say, the tip of the iceberg, and it just sort of fits in with all the other research that's been done that points to a vast number of perhaps individual triggers depending on the patient. So it sounds like such an important first step. And I know you were very gracious in thanking Propel Cure for helping to sponsor this important research. Can you just tell us more what this particular donation meant to this project and how you see Propel Cure's role in the IBD research community? That would be great.
Yeah, absolutely. I think the reason I am very grateful to have received this funding from Propel Cure is because these technologies are very, costly. These sequencing approaches for really in detail characterizing these T cell receptors in details and all the individual HLA molecules of patients were very costly techniques. And before the funding of Propel Cure, we simply did not have the funds to do this, while we really believe that this is a necessary step. And with the current funding climate, it is increasingly more difficult for researchers to do these type of necessary experiments. And therefore, this study would not have been possible, especially maybe I would have wanted to do this anyway, one way or the other, but Propel Cure made it possible that we can really do this now and accelerate the research so that we can move forward. So I think it really boils down to the costly experiments that were needed to do this were funded by Propel Cure.
And I know all the donors would agree with me that this has been so worth it because you've given us yet another increasing insight into what may be in play in IBD. And we appreciate so much having you here today, joining us, Dr. Van Unen, and sharing all your research. Thank you so much.
Thank you, Angela. It was really great to be here. I'm very appreciative.